Hi everyone, it's been a while, but in today's video I'm going to walk you through one of the most classic and powerful histological techniques we use to evaluate cartilage-like tissues. If you're working on cartilage scaffolds, chondrocyte cultures, or differentiation of stem cells into cartilage, you've probably heard of Safranin O. Safranin O is a dye that lets us visually assess how rich our tissue is in cartilage-specific matrix, especially the glycosaminoglycans, or GAGs, crucial element in ECM, in cartilage ECM, that give cartilage its structure and function. In other words, when this stain works well, you don't just get a pretty red-orange image. You get evidence of how successful your cartilage engineering strategy actually is. Safranin O staining honestly has a very streamlined and simple process. You should have already your samples fixated with formaldehyde, and depends on your case, depends on your sample number. Mainly and generally in the most cases for master projects or even PhD projects, you don't need more than 20 grams or 25 grams Safranin O. And in addition to that, you need some common solutions and materials. Safranin O agent, of course, PBS, distilled water, acetic acid, glycerol. And as labware or equipment, you need plates or dishes, cover slips, pipettes or syringes. There is no difference between them in terms of use in this process. Of course, gloves and a bright field microscope. And also your samples. Your samples could be fabricated through gelations, hydrogel gelation or electrospinning or anything else that could be able to be under light microscopy. I recommend it to use a microscope that is connected to a system or has its special camera. In my case, I use Electrospun scaffolds and chondrocytes. And after fixation of cells on the scaffolds, I bring them into the staining process. In my case, I have some scaffolds that are cell-loaded, I have some scaffolds that are cell-free, and I have some cells without scaffolds. I have samples and control negative and control positive. Now, of course, any of the cell-loaded scaffold has a different composition with different fibers or materials. That doesn't really correlate to this video, but you may have some different ratios of various materials that have different effect on producing GAGs. So you can validate that increasing a certain material can help the production of cartilage-like matrix. The first step is a PBS rinse for about 4 minutes. Just add enough PBS to fully cover the sample. No strong agitation. After that, quickly switch to distilled water for about 30 to 60 seconds. The idea here is to move the sample into a lower ionic strength solution, which helps the Safranin O bind more effectively to the negatively charged glycosaminoglycan later. Next is the main staining step. Make sure you use solution about 2 to 4 minutes. Areas rich in cartilage-like matrix will gradually turn to red-orange color. Once the staining time is up, briefly rinse to remove excess unbound dye. Quickly add distilled water for about 10 to 20 seconds. After the quick rinse, move to a differentiation step to sharpen the contrast. Immerse in 1% acetic acid for just 5 to 10 seconds. This differentiation step helps remove more non-specific background and improves the contrast between matrix-rich regions and areas that are poor in glycosaminoglycans, so your structural differences become more obvious under the microscope, not a heavy red background on your images. After acetic acid solution, very dilute acetic acid solution, we're ready to mount the samples for imaging. Gently blunt around the edges of the sample or scaffold, and in this process you can add a small drop of aqueous mounting medium, or you can use a 1-to-1 mixture of glycerol and PBS as a simple mounting solution, and the whole process should repeat for all samples. I have also some practical notes that are also important to remember. First of all, if your samples look overstained, everything is just a dark blob of red, something like that, You can reduce the staining time or even drop the Safranin O concentration to 0.05%. If staining looks uneven, try increasing the volume of stain and make sure you have gentle rocking so the dye can access all sides of the construct. The other point is for non-cross-linked alginate or similar soft hydrogels, keep all soaks short. Otherwise, you might start losing material or changing the structure. And also, once you've stained whole mount samples like this, don't run them through alcohol or xylene. That's more for paraffin sections. Keep in mind that the concentration of Safranin O in water can vary depending entirely on your scaffolds. You should choose the concentration that gives you best images. The concentration value is not a fixed, unchangeable number that is well established in all articles or textbooks or handbooks. No, this is not a firm protocol. The only important point is that whatever concentration you choose, you must keep it the same for all of your samples so that they remain comparable. Another issue is, after staining, how long do we have before imaging under the microscope? I recommend imaging as soon as possible, ideally right away. For example, my stained and mounted Electrospun samples became jelly-like and deformed after about a few hours. They were no longer suitable for imaging. If you have any questions about any step, let me know in the comments. Thanks for watching.